All, we will move to the next unit, that is genetics and evolution, that is heredity and evolution. So this chapter is very much important in the point of CBSE, and you will get a lot of questions from this. Almost 17 marks, 17 marks question, 14 to 17 mark questions alone come from this single unit, which is the two units. 5th and 6th will be of 100% genetics, and 7th unit is of evolution. We are going to study about the theory of evolution, different types of evolution, those things we are going to study. So this chapter is very important. If we understood this chapter, the upcoming chapter, that is biotechnology, will be very easy to understand. And it is this chapter is a compulsory chapter considered to the examination point of view, and there are some mathematical questions also possible from this. So each and every term is important considered to this chapter. So first we will start with what is genetics. So before entering to this chapter, in this chapter we will be studying much about the first year. For 5th chapter we will be studying about the Mendealian genetics, non-Mendealian genetics, chromosomes, and some abnormalities related to the chromosomes. If the chromosome number is added, deleted, structural variations in the chromosome, minor like that. And the second chapter, that is 6th chapter, it is 100% made upon the molecular mechanism, that is DNA. What is DNA? How the DNA replicates? How proteins are synthesis? What are the importance of proteins? Those mechanisms we are studying. So, and in the last chapter related to that, how these mechanisms are supporting for evolution. This is the content what you are going to study. So we will start from the very basic. There are some terms and terminologies are there. If you are not understood the terms and terminologies, it is very difficult to understand this chapter. So terms and terminologies, what they mean, what is the importance of that, those things are very much important. So I recommend, since we are running in this lockdown period and with schools, we don't know when the government is saying to completely open the school, we also don't know. So I can understand your situations, but I recommend you to go online and see some pictures, colored pictures. As much as possible I did, but there are many things to read, many things to understand, which is not much explained in NCERT, but it will help you very much to understand this chapter. But once you understood this chapter, it is very interesting. This genetics is very much interesting compared to other chapters, because other chapters we are running maximum with theory parts. There is less practicals. That means nothing to do much. You have to understand the facts, and that is it. If I am saying this hormone will secrete during this time, yeah, this hormone will secrete. But this chapter is not like that. So we have a lot of interesting stuffs are coming up. So we will go one by one, and plus we have to speed up also. Since it is the beginning, I will be focusing much on the basic things, terms and terminologies, what they are, and little bit of Mendealian genetics, and a small calculation about gametes. So if different types of gametes, how they are formed, then the Punnett square, and few calculation. Apart from that, how to find out the genotype, how to find out the phenotype, those things. So we will go one by one. So the term genetics, people were known about this particular thing from very beginning. It is not a new thing. Only after 1900s only it has become a big important topic, or a hot topic in the field of science, and especially in the field of biology. And it doesn't mean that before that there is no genetics. There was genetics, but people were not known much about this. One of the main reason was one of the main reason that me led this discovery of genetics, the field of genetics, was microscopes, techniques, and other stuffs that lead this genetics field to move much. And now we are in the 21st century. We are there in a position on a gene therapy. We can pick out one gene, unwanted gene, and in that position we can keep one useful gene. So we are in that level. So all of them was begun in somewhere in 15th and 16th century. But people were known everything, but it was a misconception, because some of the misconception I will tell you. Earlier times people believe like that when a new generation comes, now we know that it is a fertilization, it is the fusion of sperm and ova, but the people were not believed like that in the earlier times. People believed in such a way that inside the mother, it is one among the misconception what they have believed. They believe like that inside the mother's womb there is a mini human, which we call it as homunculus. When we are studying about evolution, we will discuss about that, but to understand, it comes in genetics also, homunculus. And when the sperm comes and it become a complete human, that is a fetus. In the modern terms we call it a fetus. This was the one belief. And the second belief was, whenever the gametes are formed, how the gametes are formed, now we know that the gametes are the genetic, that is especially the chromosomes, they are forming, that they are segregated properly in both male and female through the process of mitosis and meiosis, which leads to the formation of gametes, ovum and sperm, that we know now at present. But earlier people believed that inside the gametes there is a small part of head, there will be a small part from the hand, a small part from the leg, a small part from the stomach, intestine, eyes. Everything will be put inside one gamete and others will be put in another gamete. When these two fuses, it will give a fetus. So these type of theories were in existence and the people believed also. And many of the scientists, like at the time of Louis Pasteur, these are times when the Louis Pasteur, Francisco Redi, those scientists were lived. Even they strongly opposed this. And that time the people even believed in theory of spontaneous generation. Spontaneous generation means the life will form from nothing. That means if we keep a sack of rice to the corner of a wall, next day you will see mice or rat or some type of insects. So they believed in that theory of spontaneous generation. So these all things were questioned by the studies which was put forward through the genetics. And genetics means, the term genetics or gene or whatever it is means your existence, that means your beginning, that it was originated from a term known as genesis, which means origin, and from that term the genetics came to existence. And in biology, the genetics, biological term, it is 100% related to the genetic, related to the genes and the heredity of the organism. So one of the most important person who led the studies in genetics was Mendel. But whatever the Mendel has studied, Mendel was in the time of 1850s, that is in the 18th century. Mendel was, whatever the studies the Mendel was put forward, it was not ready to be accepted by the scientific community of that time. They said that what Mendel is saying, it is not at all possible, and they rejected the theories of Mendel. Actually, even though the Mendel was a biologist, but actually the Mendel was not 100% biologist. He was a good mathematician. Whatever the predictions he did, he used the terms and the basic concepts of mathematics. So he was a very good mathematician, especially statistician. statistics, that is the most good term he was used. And in 1900s, that means after 1920s, after the First World War, beginning or after the First World War, these biological studies started to become something like a core to the scientific realm because of the microscopes, techniques and technologies, and chromosomes were come to know, nucleus we came to know. That time three of the scientists, when they were formulating the chromosome, that means the chromosomes are responsible for this heredity and variation, that means changes from the parents to the young generation. They found out that very before than them, there is one person who came to know about this thing. He published it, but it was not accepted by the scientific community, so there was no importance for that, for his theory. So his theory was rejected by those scientific community. From there onwards, these three scientists, we will discuss about them in these scientists, that is a chromosomal theory of inheritance. We will study about them, these scientists, and what they did and how it is. They went back and took whatever the Mendel has studied and they added it to their studies, and they made Mendel as the father of genetics. So that is why still we believe that, not believe, it is 100% sure that Mendel or Father Gregor John Mendel, he was a monk, not nun, he was a monk. So Father Gregor John Mendel is considered as the father of genetics. And when they started to study about the things, Mendel introduced many things, that LMND, when he was studying, he found out that the substance that moves from one generation to a generation, the Mendel actually called it as LMND, and other person called it as factor. So it was very difficult to put these terms into a scientific research paper. So those terms were redefined. They gave a name and meaning for them. So we will first see those terms and terminologies, what they have given. So in the terminology, the first thing that comes is gene. Gene means same thing, origin, from where you are origin. So what is a gene? Gene is a short segment of DNA that is responsible for expressing a particular character. That means a small stretch which have the capacity to express a small stretch of DNA which have the specific capacity to express a character. That's all. And the Mendel was the person called these genes are elements, and Carl Correns, he called it as factors. After that, in 1909, these gene was introduced into the system of biology, and the term gene was given by Wilhelm Johnson in 1909. Then comes what is a genome. It is a total genetic composition of an organism. So what are the that means all the genetic things that is present inside a organism is called as genomes. Next one comes is alleles. The term alleles means an alternative form of gene, alternative form of same gene. For example, when we are taking a character, for example, the best thing what we have all studied is the height. Height can be again divided into two parts. One is tall, another one is short. That particular alternative thing which focus to one of the character is called as an allele. And if you see that near to that, there is one diagram also is given, which is a structure of a chromosome. And if you see that, you can see something written as A, capital A, capital A, capital A, capital A, small a, capital A, and in the top you can see that A, capital A, small a, allele 1, allele 2. So these two alleles, one allele comes from the father and another allele comes from the mother. So these two alleles are responsible when, if the father's allele is A and the mother's allele is small a, the child may express any one of these alleles, either capital or small. So they are known as the alleles. And the location or position of that allele in a chromosome, we will call it as locus. That means inside a chromosome where these alleles are located or positioned, we will call it as locus. And in some cases these alleles may express, maybe it may express capital A, capital A. If it is expressing the same character or same form, capital A and capital A, we will call it as homozygous. If one chromosome expressed A, one sister chromosome expressed A, and another sister chromosome expressed small a, that means two different character or two different forms which represents to a character is expressed, we will call it as heterozygous. So these terms are very important, that is capital A, small a. And what is known as a dominant? If it is capital A and capital A, for example a height, if inside of me the height, for a height character, if from my father and from my mother, which gene has come? If my father is very tall and mother is also very tall, so there is a chance that I may get both the from parental side as height as TT, which is homozygous, and I will be tall. That is a consideration of Mendel. So which one is expressing that particular allele is known as a dominant allele, and which one is not expressed is known as the recessive allele. And then we comes the genotype. Genotype is a genetic expression of an organism. That means inside the gene how it is, whether it is capital T or capital T small t, however it is, that particular condition or that particular type is known as genotype. And there is one more thing which is known as the phenotype. Phenotype means express outside. For example, inside of me there may be a gene for capital T also, that means for a height, tallness, and there may be a gene for small t also. But if capital T is expressed, that one we call it as phenotype. That means which I can see from outside, that particular thing is called as phenotype. And then comes the character means general feature of an organism. How can I generalize it, that particular organism is tall, this particular organism is short? So these are called as characters. And other one which we call it as monohybrid, we will study about that. Monohybrid cross, dihybrid cross, we are coming, polyhybrid cross, we are coming to that. Then comes the filial. Filial is very simple, it's a German term which we call it as generations. Parental generation, that is the P generation, then F1 generation, F2 generation, very simple, they are called as offsprings. Then pure line means which is as parents are the same genotype and phenotype is pass over through one generation to another, that particular condition is called as pure line. And test cross and back cross, we will study about that. And if you see that inside an organism, for example inside a human being, our chromosomes are arranged like this: chromosome 1, 2, 3, 4, 5, 6, 7, 8, like that we are having totally 23 pairs of chromosome, that is 46 in number. 23 multiply with 2, which is called as 46 in number. And this arrangement of chromosome according to its number, according to its height, we call it as karyotyping. And we can take one chromosome and I can study it very specially. That means which gene, where, how. That is called as ideograms. So whenever we are doing this ideogram, chromosome we will study like this, and each the chromosome will be having two arms. The upper one is known as the P arm and the down one is known as the Q arm, and wherever the two chromatids are joined we will call it as the centromere. And whatever the light stated and dark stained, they are all different types of gene and its position 15.33, 15.32, they are all how these chromosomes are. What are the different types of genes located in each chromosome, and studying like this we'll call it as the ideograms. And next one we will move to the Mendealian genetics. That means very straightforward, we'll move to the Mendealian genetics. And in Mendel, when he was doing his studies, Mendel was very specially he selected some things, experimental material. Since Mendel was a priest, so he was living inside a very normal area, and he will be living inside a monastery, so he can't go out and do some other experiments. So he chose his experimental material as pea plant. Why he chose pea plant? Because since he was in Europe, he was in somewhere in Bonia, present day Czechoslovakian border of Czechoslovakia. So since he was there, pea was a common food that the foreigners use as a lunch and dinner. So they are very much familiar with these particular plant. And one more additional thing he was found is its short life. That means the lifespan of a pea plant is only for 6 months. So within 6 months it will produce the offspring, and it will produce n number of offspring. And whenever the pea plant is when next generation comes, there are it comes a lot of character wise. That means some of the pea plant it is having a violet color flower, some will be having white color flower, and if you see the pods are of different shapes, and if you see the size of the pea, it may be round, it will be wrinkled. So lot of characters Mendel got there to study. And Mendel was a very strict botanist also the same way. And what Mendel did is, Mendel has chosen the material pea plant and he did artificial hybridization. Artificial hybridization means pea plant normally they do self-fertilization. Since they do self-fertilization, which type of characters and which type of pollen grains falls there nobody knows, and what are the next type is going to come we can't predict. So Mendel did it in a controlled area. That means in the said textbook they have given that Mendel has taken a flower and he first cut the stamen of that flower, and he chose a particular stamen, that is particular pollen grain, and he self hybridized it. That means artificially hybridized it and covered that particular flower, and he waited for the next generation. What is the characters it is going to come? So whatever the experimental materials we do, the best important thing is to choose with very simple things. So first Mendel decided to take single character, then he decided to go for the 2 or 3 characters like that. So what Mendel did is, Mendel has chose the flower. He did the same technique, that means the artificial hybridization technique he has followed. That means a stamen will be artificially removed, then the interested which part of the flower's pollen grain is interested from the pea plant, he will dust on it and he will follow the bagging technique and he will wait for the next generation. So first he put his focus in a single character. For his single character, what he did is, he choose, as per the textbook, if you are going, he choose the character tall and tall. That means he chose the height of the plant, and here I have given the color of the flower. So both are same, the result will be same. So here when he chose that, when Mendel started to do that, since he has used only single character and he is doing the process of hybridization, so he called it as monohybrid cross, monohybrid crossing. So here what he did is, in the parental character he chose a flower which is violet, it's common, and he chose another flower which comes in the pea plant which is white. And if you are going according to the textbook, he chose the character of height, that is one tall and another one short or dwarf. So he crossed it. In the F1 generation, he got all the plant or tall. As per here, all the colors violet, flower color was violet. So he was expecting, he may be expecting something like that. It should be a mixed color, or few will be violet and few will be white, like that. But he got all the first generation as F1 generation as violet, and then he self-crossed the F1 generation. In the F2 generation, he got an ratio that is 3:1. 3 flower which is of violet color and 1 flower which is of white color, or as per the textbook, 3 tall and 1 dwarf, he got it. So he was thinking, why like this? Why these type of things are happening inside the plants? So if in the F1 generation all are violet, in F2 generation all should be violet. Why I am getting white? So he understand that there is something inside this plant which carries, that means there is an element A inside. That's why he called it as element A or factors. There are some factors inside the plant which are responsible for both the characters, that means violet and white. So in order to understand this, what he did is, he did the test crossing. Why he did the test crossing? Because he got one flower as white and all others as violet. So in order to understand whether the parental generation is also carrying the same, so what he did is, he took the recessive phenotype, that is the small p, crossed with the dominant phenotype, that is capital P, and he got that all the F2 generations are, maximum of the F2 generations are heterozygous in condition. That means it carried both the alleles for capital P and small p. That is why in F1 generation it has having all as heterozygous. That is why in the F2 generation they got the one as violet color and another one as, all others are violet color and one as pink or white color he got. So he called those factors as element A. From this he came to one theory also. The theory is this: the characters can segregate. That means characters can divide according to their need. It is not depend upon the environment, or it is not depend upon the fact or the external things. It can segregate according to their need. And one more thing, he also confirmed that among the parental generation, one among the character will be dominant. So he called it as, so he proposed two laws. One is law of segregation and another one is law of dominance. So two laws he did. After doing this, he chose this. I was copied from the Byju's because of the illustrated color illustration. He chose dihybrid. Dihybrid means whether the same 3:1 ratio will follow if there is choosing two characters at a time. But when he was choosing the two characters at a time, he followed the same technique. So he used the seed color, seed. One seed he used as yellow and round, crossed with green and wrinkled. He got yellow round in the first generation. He self crossed it and he got a ratio that is 9:3:3:1. 9:3:3:1. It was a totally different thing from the first experiment. What he did is in the monohybrid cross. So he said that, and from that he came to another thing, that theory of independent. That means he came to the third law, that is independent assortment, theory of independent assortment, or law of independent assortment. That means each character can divide according to their choice. It is not a thing that yellow every time it should be yellow round and every time it should be green and wrinkled. No, characters will segregate, characters will assort, or that means it will mix as per their need. That he called as the law of independent assortment. Independently it will do, it is not depend upon anything. So these two things he came from that. Two laws, three laws he put. And in order to study these Mendealian laws, one scientist known as the Punnett, he developed a technique called the Punnett square. And if you see here, this Punnett square, you can use any character as your wish. But since I copied this from the net, so I have used the character of mice. That means one mice which is brown in color and another mice which is white in color. So one mice is homozygous, one is heterozygous, and another one is homozygous recessive. Heterozygous dominant and homozygous recessive. So when they are crossing, so it is very easy. That means the one part of the A will be crossed with the heterozygous, and they will get four offsprings they will get. Among that four offsprings, two offsprings will be brown in color and another offspring will be white in color. So this what can be done by using the Punnett square. So it is very easy to do any of the characters. Apart from that, a technique which is already determined, that is the test cross. And why this test cross is done? Because in order to find out the genotype of an unknown parent, that means purple flowered parent, any color, that means capital P or capital B, crossed with the white flower, small b. So if they don't know whether the parent is a homozygous or heterozygous, when they cross this with a dominant phenotype,